Good morning to all of you. In spite of the heavy rain and all the constrainment, you have, at least few of you have turned up to this online class, and I hope others will join. Maybe some of your classmates or friends may be having problem with power, electricity, or this telephone communication system because of the heavy rain. Afterwards, however, I am putting that in the YouTube channel where they can download. So today, after completing the nervous system, we will start the new topic that's about the muscle contraction. It's a very, very interesting topic to know. In our daily life, we carry out lot of activities. Mainly, I am talking about the physical activities. Either we may be running, moving, walking, eating, exercising. We carry out lot of activities. How we are able to carry out these activities? We say that okay, if I have to walk, our legs will move. If I have to do something with the hands will swing. Now the question is, what makes our legs or hands to move? How the movement or the locomotion is brought about? Different organisms carry out this type of activities, including the locomotion, by different means. But whatever may be the means of these physical activities or locomotion, that is carried out with the help of what we call our muscular system or the muscles. Even there are certain muscles which are working continuously, certain muscles which are working according to our will and wish. In the sense, for example, if you take the heart, which pumps the blood continuously since the day we take birth, even before coming out of the uterus of the mother, the heart has started its working, and it will continue to work till the last breath. That means this is a continuous process. Again, it is also achieved by the muscles. So that's
Sir,
why, yes, ಹೇಳಿ
ಸ್ಲೈಡ್ ಶೋದಲ್ಲಿ ಗೂಗಲ್ ಮೀಟ್ ವಿಂಡೋ ಓಪನ್ ಹಾಕೊಂಡು ಉಂಟು ಸರ್.
ಇಲ್ಲ ಇಲ್ಲ ನಾನು ಇನ್ನು
ಸ್ಕ್ರೀನ್
ಸ್ಲೈಡ್
ಶಾಟ್
ಶೋ
ಅಲ್ಲಿ.
ಹಾಕೇ ಇಲ್ಲ.
ಹೌದು ಸರ್.
ಹಾ
ಓಕೆ.
ಓಕೆ, ಈಗ ನಾನು ಬರ್ತಾ ಇದ್ದೇನೆ. ಓಕೆ, ನಾವು ಐ ವಿಲ್ ಪ್ರೆಸೆಂಟ್ ದ ಸ್ಲೈಡ್ ಶೋ. ಈಗ ಕಾಣಿಸ್ತಾ ಇರ್ಬೇಕಲ್ಲ ಸ್ಲೈಡ್ ಶೋ.
ಹಾ ಸರ್, ಕಾಣಿಸ್ತಾ ಉಂಟು.
ಹಾ, ನಾನು ಇಷ್ಟೊತ್ತು ಸ್ಲೈಡ್ ಶೋ ಹಾಕೇ ಇಲ್ಲ. ಸೊ
Yes, sir.
the muscular system or muscle contraction, I will be discussing with you under the heading locomotion and movement, because these two things are same. So here, when I talk about the movement, there are basically three types of movements, and these three types of movements have evolved over a long period of time. What are these three types of movements? Amoeboid movement, ciliary movement, or and the muscular. Probably, being the student, you remember what you have learnt in your first semester, BSc first semester. If you have not, just recollect. What do you mean by this amoeboid movement? Amoeboid movement is a specialized movement where it is shown by some of the unicellular or acellular organisms using the pseudopodia. By using the pseudopodia, they achieve the movement. And in the same class, there are many other organisms in the same phylum which undertake the movement using either cilia or flagella. So ultimately, their aim is to undertake the movement. So this is achieved by either cilia or flagella. But if you come to the higher organ, this movement or all our physical activities are because of our muscular movement. So here we are mainly concentrating on our muscular movement. How these muscular movements are brought about? The muscular movements are brought about by the muscles. These muscles, most of them or large quantity of these muscles are attached to our bones. How the muscles can attach themselves to the bone? There is an attachment what is known as tendon. Tendon is the one which attaches muscles to the bone. And the muscle work in antagonistic pairs. What do you mean by this antagonistic pair? Some muscles responsible for contraction, some for relaxation. And some muscle contract while other muscles relax. So all our physical activities, including the movement, is brought about by the muscles. Whether we have only one type of muscle or all muscles are uniform, no. Even in muscles also we have diversity. Basically, we can say that there are three types of muscle tissues: skeletal muscles, smooth muscles, and the cardiac muscles. What do you mean by the skeletal muscles? The muscles which are attached to our skeletal system, that is the bones and cartilage, we call it as skeletal muscles. The second category of the muscles is known as the smooth muscles. These are the muscles which are associated with our tubular organs. For example, our entire digestive system is also lined by the muscles, and those muscles are known as smooth muscles. And the last one is the cardiac muscles, which are lining the wall of the heart and responsible for heartbeat. So there are three basic types of muscles, and each muscle has different characteristic, structure, and shape. Here I have shown you some of the muscles photographs. By seeing the photograph only, you can differentiate these muscles histologically. But we will discuss about these muscles in detail. I have told you the major portion of the muscles are nothing but the skeletal muscles. These skeletal muscles, they are also called as striated because they show alternate light and dark banding and thus appears striations or show striations. They are also voluntary muscles. What do you mean by these voluntary muscles? They function according to our will and wish. Suppose if you want to stretch your hand, if you wish, you can stretch. If you can fold it, you can fold. That means that works under your control. Voluntary. Then they are multi-nucleated. In a single muscle fiber, you can expect more than one nuclei. So that's why it is multi-nucleated. And their shape is also, as they are attached to the bones, they are long, thin, and mostly cylindrical and responsible for the movement of our skeleton. So this each muscle cell is commonly called as muscle fiber. Why? They are long and thin and cylindrical. They appear like fiber. So that's why they are called as muscle fiber. One muscle fiber refers to one muscle cell. And the skeletal muscles, they do not divide to create new cells. Remember, one muscle fiber cannot regenerate or give birth to another muscle fiber. And new fibers, then you may ask how these new muscle fibers are formed. New muscle fibers are produced by the stem cell. What do you mean by stem cell? These are the cells which are produced in our bone marrow where they are not differentiated. And stem cells are the basic cells. From that, you can form any type of cell. So, these stem cells develop into skeletal cells or muscle fibers. But a muscle fiber cannot give rise to a muscle fiber. And as I have told you, they are striated because there are cross stripes, and these cross stripes are popularly called as bands, and they are perpendicular to the cells. Probably here you can see the photograph. In the photograph, you can see the muscle fiber. See, these are the muscle fiber, and they are multinucleated, where you can see, and you can see the striations in all these things. Can you make out the striations? Some dark area, some light area. These are all stained ones, so that's why it appears clearly. In all these, these are all the muscle, skeletal muscle tissues or skeletal muscle, where one muscle fiber has been highlighted, and that shows these striations, cross striations, which are perpendicular to the fiber. Then the next one is, there is somebody. Next one is the cardiac muscle cells. The cardiac muscle cells are popularly called as cardocytes. They are mainly found in the myocardium layer of the heart. They are found in the wall of the heart, and they are branching cells. They show branching pattern, and they are connected with the other fiber with the help of intercalated discs. As a result, the contraction will be very fast. Contraction will be very fast. Continue. 1, 2, 3, 4, 5, 6, 7. Branching, connecting at the intercalated discs that allow the contraction to occur fast and at regular, and they are regulated by the beating, the contraction that has been achieved by the cardiac muscles. It is achieved by some specialized cells, what we call pacemaker cells. These are not the muscle cells, these are the nerve cells. They regulate the contraction of these heart muscles. So, the contraction of the heart muscle is brought about by or regulated by the pacemaker cell, which are nerve cells. Remember, these cardiac muscles are also striated, but you can see faint striations. But they are involuntary. You cannot ask your heart muscle to stop or to function. It is involuntary. As I have told you, they started contraction from the inception of our life in the uterus of the mother till the last breath. So, and they are uninucleated. They are having single cells. See, these are the cardiac muscles where you can see the branching. These are known as intercalated discs. Intercalated discs, they branch and join. And striations are also there. And this is how the cardiac muscle tissues appear under the microscope. Then the last one is the smooth muscle cells. All, all are small muscles and they are pointed at both ends. Small muscle cells are pointed at both ends, can divide and regenerate new cells. This is the specialty. Smooth muscle cells can divide and regenerate the new muscle fiber. They are also no, but they are non-striated. No striations are seen, and they are involuntary. They are not under the will and wish of us, and they are having single nucleus. Where do you find them? They are found lining the hollow organs, entire our alimentary canal, respiratory tube, even in the intestine, bladder, lungs, all blood vessels. The smooth muscles are there, and they bring about a rhythmic contraction by means of which they can move the substance through these hollow organs. For example, what happens if you eat something You swallow it, and then it moves along your intestine, along with your alimentary canal, gastrointestinal tract. How they move? Do you think they are moving only with the gravitational force? So you are sitting upright. So whatever the thing that you put in mouth, it goes to your stomach and intestine. Even when you are sleeping, also they will move. The food will move. How? This is brought about by the rhythmic contraction exhibited by the smooth muscles. They squeeze the things through uh, the tubes. It is just like if you squeeze the toothpaste, how the paste moves out. So in the same fashion, the rhythmic contraction of the smooth muscle helps in movement of the many of the things, including the blood, through this hollow organs. See the smooth muscles where there are no striations, and each one, if you see, they are having pointed ends. See, all of them are having pointed ends, and here one muscle fiber I have exhibited where I am moving the cursor. They are having centrally located nucleus and tapering towards both the ends and thereby making pointed ends. So these are the three different types of muscles, and these three types of muscles together carry out all our vital activities, which is very essential for the survival of any living being. So, this is the differences. Skeletal muscles, they are striated and they are multinucleated and they are voluntary in nature. They function according to will and wish. The cardiac muscles are also striated, but they are uninucleated and they are involuntary. Whereas the last one, smooth muscles, they do not have any striations, single uninucleate and involuntary in nature. So now, as far as our study is concerned, we will be concentrating more on the skeletal muscles because these are the muscles uh, which is found in large quantity and responsible for most of our physical. For example, human body contains over 400 different skeletal muscles, each having numerous muscle fibers. There are in our body over 400 muscles. They contribute around 40 to 50% of our body weight. If you are muscular, your weight will be more. When I say a person is muscular, he is having very strong muscles. So that contributes. The skeletal muscles, which are attached to our bones, are responsible for almost 50% of our body weight, 40 to 50%. These skeletal muscles, they have a lot of functions to carry. One is locomotion or breathing. How? By force production or whatever the posture that we show, postural support. Again, because of skeletal muscles. Heat production during the cold stress. Probably, at least all of you might have understood that. What happens if you experience very cold or chill? Immediately your muscles start shivering. You start shivering, isn't it? What is shivering? The it is the mechanism by means the skeletal muscles are producing more and more heat because you are exposed to cold stress. So that's why the skeletal muscles not only responsible for the locomotion, breathing, postural support, they are also responsible for heat production even when we are exposed to cold stress. If you see the structure of skeletal muscle has three layers. Outer, these muscles are found in bundles. Muscles are found in bundles. Epimysium, which is the layer or a membrane which surround the entire muscle. There are 400 different types of muscle. Each muscle is surrounded by externally a membrane or a layer, what is known as epimysium. And within this muscles, you can see the bundles, muscle fibers, which are bundled together. And each bundle is surrounded by another layer, what is called as perimysium. And within the bundle, if you look into the individual muscle fibers or individual muscle cells, they are surrounded by endomysium. See, here you can see this is a entire, this is the muscle of biceps, what you call biceps muscle. See, externally the entire muscle is surrounded by epimysium. Epimysium. And this epimysium is inside which you can have the muscle bundle. See, 1, 2, 3, 4, 5, 6, 7 muscle bundles you can see. And these bundles are covered by perimysium. And inside, in each bundle, there are numerous fibers, what we call muscle fibers, which are nothing but the individual cells. They are surrounded by endomysium. So, muscle bundle, see, this is surrounded by perimysium, and individual fiber, it is being surrounded by endomysium. Each muscle fiber, if you observe, this has been covered by a plasma membrane or cell membrane. Cell membrane or the plasma membrane of the muscle, it is known as sarcolemma. The in a normal cell, you call it as plasma lemma or plasma membrane, whereas in the muscles, the same is called by sarcolemma. Now, each muscle fiber, if you have, if you look into, they are having thread-like strands within the muscle fibers. Within a single muscle fiber, there are innumerable number of thread-like strands, and they are called as myofibrils. Myofibrils, which are made up of protein. There are two types of myofibrils. One is the actin, which is also known as a thin filament made up of tropionin and tropomyosin. These are the proteins. And another one is the myosin, which is a thick filament. So, in short, if I want to say, within the uh, muscle fiber, there are thread-like strands, which are called as, which are made up of actin and myosin, or which are also named as actin filament and myosin filament. Actin filaments are known as thin filaments, and myosins are known as thick filaments. So here, the each muscle fiber within a bundle, it behaves as a single unit. Each muscle fiber, if you see, it behaves as a single unit, and it has multinucleate. Already told you, I have told you, the skeletal muscles are multinucleate, and they are made up of myofibrils. What are those myofibrils? Actin and myosin. And these myofibrils are surrounded by sarcoplasmic reticulum. What is this sarcoplasmic reticulum? Endoplasmic reticulum of the muscle is known as sarcoplasmic reticulum. Sarco refers to muscles. So, sarcoplasmic reticulum is nothing but the endoplasmic reticulum of the muscle fiber. And these myofibrils are invaginated by a tubule, that is the transverse tubules. In short, they are called as T tubules. transverse tubules, in short, which are known as T tubules. Each myofibril, if you see, contains interdigitating, which are alternately placed thick and thin filaments. Thick and thin filaments. Thick filaments are myosin filaments, thin filaments are actin filaments, which are arranged longitudinally and cross-sectionally also. Longitudinally and cross-sectionally also they are seen. There is alternately, you can see that the thick and thin filaments are arranged, which you can see here. Now, I have tried to enlarge one single muscle fiber to understand the organization of the muscle fiber. See, a single muscle fiber, it has two ends, and that is known as the, here see the Z line or Z disc and Z disc, which you can see here. Here also you can see that whatever the things you see in between two adjacent Z discs, they are known as sarcomeres. I will come to that later again. And here within this we have two regions. One is the I band and another one is the A band. I will tell you what they are afterwards. And here you can see the thick and thin filaments are arranged alternately. Thin, thick, thin, like that. They are arranged longitudinally and alternately. And here, see here, the thick and thin filaments in the lower diagram you can see, and thin filaments and the thick filaments. So a muscle, this is the entire muscle. Within that there's a bundles of fibers. If you enlarge a bundle, within that you can see a single muscle fiber, and within a single muscle fiber we have myofibrils showing light and dark bands. And this is also another structure for single muscle fiber, where you can see, just observe, I will explain later what are these I band, A band, Z line. You must see that you may have the confusion. See, this is the single muscle fiber, and this single muscle fiber, you can see there's a sarcolemma membrane and nucleus and the mitochondria and then the tubules. What are those tubules? Actin and myosin tubules. A I have told you, muscle myofibril is nothing but a single muscle fiber. It has Z line, A band, and I band. Within the sarcoplasm, what is sarcoplasm? The cytoplasm of the muscles is known as sarcoplasm. Within the sarcoplasm, that is the cytoplasm of the muscle, you can see sarcoplasmic reticulum. That is the endoplasmic reticulum of the muscle for the storage of calcium. Then it is also having transverse tubules, which are known as T tubules, and terminal cisternae. See the structure of the muscle tissue again. This is the sarcomere. This is the one sarcomere. Within a fiber, you can have a number of such sarcomeres. What is the sarcomere? The distance between two adjacent Z lines. Then there is the myofibril, muscle fiber, and all those things. Sarcomeres, why the sarcomeres are important is They are the basic contractile unit because for any physical activity there must be contraction of the muscles as well as the relaxation. So this contraction is brought about by the individual units of this muscle fiber, what is known as sarcomere. So now, if you observe the electron microscopic studies or electron microscope photographs, you can see that the myofibrils are made up of repeating dark and light bands, which you can see clearly. The middle of the dark band, you can see what is known as the M line. The middle of the sorry, the middle of the dark band, we have M line, and in the middle of the light line, you have Z line. In the middle of the light line, you have Z line. So the repeating units of one Z line to the next is called sarcomere. See, one sarcomere means this is the Z line which is found in the light band. Then it is followed by a black or a dark band. Then again a light band. So in each light band in the center you have a Z line. This repeating unit from one Z line to next, it is called as the sarcomeres, and they are responsible for bringing the contraction. So sarcomeres are the contractile apparatus. So the muscle cells are also, they are muscle fibers because they are single long cells. Just to summarize what I have told just now, muscle fibers are made up of myofibrils. Myofibrils are made out of units called sarcomeres. And sarcomere is made up of thin and thick filaments. Z line is the end of the sarcomere. On both the sides, there's a Z line, and that's the end of one sarcomere. Thick and thin filaments slide over one another to contract the muscles. I will come to that. The mechanism and physiology I will discuss later. And thick and these thin filaments, they slide over one another and they shorten the muscles during the contraction. So I have also told you about A band and I band. The thick filaments produce a dark A band, anisotropic band. A means anisotropic band. It is produced by the thick filaments, that the myosin, and the thin filaments, they form the um, they are made out of mainly actin, and this thin filament extend in each direction from the Z line. Where thin and thick filaments do not overlap, this portion is known as I band, isotropic band, where thick and thin filaments do not overlap. But there's also one more zone, what is known as H zone, where thick uh, so the H zone is the portion of the A band, that is anisotropic band, where thick and thin filaments do not overlap. The entire structure of thick and thin filaments between two Z lines is called a sarcomere, and that is one contracting unit. Here you can clearly see the this is the thin line made up of actin. This is the thick line made up of myosin, and Z line C in two, where two light bands, or what we can call, where two actin filaments are joined here. Can you make out here? Z line. And M line is the one which is in the center of these thick filaments. Here we have this is the protein I zone, and here only thin filaments, and here only thick filaments. Here and here you can see overlapping the thick and thin filaments. Here only thin filaments, here only thick filaments in this region, and here you can see both thick and thin filaments. Here the one sarcomere refers to the this much between two Z lines. M line, see here, this is the one where in the center of the thick filament, and where thin and thick filaments do not overlap. That is known as H band. Whereas this is the region where only thin filaments are there, and that is called as I band. But the zone where thick and thin filaments overlap, that is known as A band, anisotropic band. This is the thick band and this is the thin band. See here also in the photograph also you can see the alternating Z line, and within Z line you can see the M line, and here you can have the thin and thick filaments. Surrounding the myofibril of each muscle cell, extensive endoplasmic reticulum, you can see the network of endoplasmic reticulum, that is known as sarcoplasmic reticulum. This network of sarcoplasmic reticulum has some facial function to do and it's very important in controlling all the activities of the muscles, especially the contraction of the muscles. See here, again I am talking, you should not confuse, this is the Z line. This is the Z line. In between that, you can have light band, you can also have dark band where they are overlapping, and see the Z lines are attached with the thin filaments. There's the actin filaments. See, this is the molecular organization of thin and thick filament. This is the thin filament. This is the thick filament. If you study the thick filaments, it is having number of proteins known as the myosin, in that the tropomyosin, which is having a club shaped head. The protein is having a club shaped head. Can you make out here? So, if you observe the very high resolution electron microscope, where you can see each myofibril is made up of parallel filaments. What are those filaments? Actin and myosin. And they are also called as thick and thin filaments. These two filaments are linked at the intervals, and those linked where they link, it is known as cross bridges, and which usually stick out from the thick filament. See, the thick filament or the myosin, where you can see myosin heads. Myosin molecule, it is having two heads. It consists of two heads. It's a myosin molecule is shaped a bit like a golf bat, and each myosin molecule having two heads. The heads usually project out from the cross bridges, or they stick out from the cross bridges. And many of such myosin filaments together they form the thick filament, having which looks like a golf club with two heads. Whereas the thin filaments, they are made up of a protein what is known as actin. Thin filaments also contain one more protein, that is the tropomyosin. Major composition of the thin filament or actin filament is the actin and tropomyosin. These proteins, both actin and tropomyosin, both are involved in the control of muscle contraction. See, actin monomers, and here they are also having tropomyosin protein. Now you observe this slide very carefully. The first photograph is the one when taken when the muscles are in a relaxed state. Relaxed state. Just see, this is the length of one sarcomere when it is in a relaxed state. The relaxed sarcomere, these are the jet lines. This is one sarcomere in a relaxed state. But below you can see it's a contracted sarcomere. What is happening is during the contraction, light bands become shorter, almost dark bands stay at the same length, but the light bands will become shorter. As a result, the total length of the sarcomere has decreased this much. So, in the relaxed state, the sarcomeres are long, whereas contracted state, the sarcomeres are short. You can see when you compare these two photographs, it is very clear. Then, before understanding this muscle contraction, we should know the role of transverse tubules and sarcoplasmic reticulum. What are these transverse tubules? The transverse tubules are nothing but the extension of sarcoplasmic sarcolemma or sarcolemmal membrane. Transverse tubules are the part of the cell membrane of the muscle, that is the sarcoplasm, sarcolemma. And in addition to covering the entire muscle fiber, these sarcolemma membranes invaginate deep into the muscle fiber, and they are known as T tubules. So, this sarcolemma membranes itself are known as T tubules, and these T tubules are responsible for carrying depolarization from the action potential at the muscle cell surface and then the interior of the fiber. Like the nerve contraction, in muscle contraction also, action potential is to be generated by the depolarization of the nerve, this muscle. So these T tubules are responsible for carrying this depolarization current, that is the action potential, from the surface of the muscle cell to the interior. Second one is sarcoplasmic reticulum. I have told you there is the endoplasmic reticulum, which is the site or the area meant for storage of calcium ions and the release of the calcium ion, because in the process of muscle contraction this calcium ions plays a very interesting role and important role. Excitation and contraction, both of them are under the influence of the release of this calcium ions. So now, coming to this muscle contraction, there are different models have been proposed in order to explain the muscle contraction. Out of them, one model is the most significant model, and it explains this muscle contraction to the maximal possible extent of belief. This is known as sliding filament model. There are two filaments which you know, actin and myosin filament. They slide over each other. When they slide over each other, that brings the muscle contraction. This is the central dogma of sliding filament model. The two filaments, light and thin and thick filaments, or actin and myosin filaments, they slide over each other, thus bringing the contraction. So during this sliding, what happens is the myosin, which is having a clubbed head, it develops cross bridges, or there is a formation of the cross bridges between the actin and myosin filament. When and with the help of these cross bridges, actin filament can slide over the myosin. This results in the reduction in the distance between the jet lines of the sarcomere. As a result of which, the muscle fiber shortens and that brings the contraction. Now the question is whether a muscle fiber can contract on its own. Yes, of course, in case of the smooth muscle fiber and cardiac muscle fiber. But majority is the skeletal muscles. Already I have told you the skeletal muscles are voluntary muscles which function according to the will and wish of the individual. So that's for this to happen. And if you wish to contract or to do any activity, it should bring ultimately or it is realized by contraction and relaxation of the muscle fiber. How this contraction and relaxation is initiated is what is known as excitation contraction coupling. Excitation contraction coupling, which refers to the mechanism. It's a mechanism where the action potential causes the myofibrils of the muscles to contract. The action potential will cause the myofibrils of the muscles to contract, thereby developing excitation contraction coupling and brings the process of muscle contraction. I don't know whether I will be able to this one, but still, though I will repeat it in the next class, but now I will give you some basics of this one. Already you have seen the muscle, the thick filament or actin muscle, actin filament, which is having a head. And these heads are known as actin binding sites. On the myosin, we have a head which is known as actin binding sites. These heads are very essential for the formation of the cross bridges between actin and myosin filaments. This site at this head, that is the actin binding site, how it binds? By the hydrolysis of ATP molecules. ATP molecules, hydrolyzing ATP molecules. For this, an enzyme that is myosin ATPase also accelerate the reaction. Both actin, myosin has actin binding sites on its head. But like that, actin is also having the myosin binding sites. When the muscles are at rest, myosin binding sites are covered by tropomyosin. See, when the muscle fiber is at rest, this is the tropomyosin. Another protein is the actin and the tropomyosin. On the actin, there are myosin binding sites. But when the muscle fiber is at rest, the actin binding sites on this thin filament, they are covered by another protein what you call troponin. So this myosin binding sites are masked by this tropomyosin, and thus myosin cannot interact with the actin. If the contraction has to happen, the tropomyosin is removed so that myosin binding sites get exposed, move out of the way, and they can bind with the myosin head. This is the myosin head. Here on the myosin head, there are actin binding sites. On the actin, we have myosin binding sites. When the actin is at rest, they are covered by this tropomyosin. But when the cell has to contract, this tropomyosin is removed and myosin binding sites get exposed and can interact with the actin binding sites of the myosin. See, the tropomyosin molecules lie on the top of this actin sites of the actin, and so that there will not be an attraction between the myosin and actin filaments when the fiber is at rest. Only during the contraction, this tropomyosin complexes are removed and actin sites get exposed. How? First, the calcium ions, they bind with troponin C, that which is nothing but a troponin complex undergo which has formed because of the conformational changes, and that moves and uncovers these active sites on the actin, thus allowing the myosin cross bridges to bridge between actin and myosin. That is what we call the cross bridges. Cross bridges will develop between actin and myosin. So see here, this is the myosin, this is the actin. They attach on the active sites on the myosin. So for the day I will stop. ಏನು ಕೇಳ್ತಾ ಇತ್ತಾ? ಹಲೋ ಕೇಳ್ತಾ ಇತ್ತಾ? Just answer me.
Yes, sir. ಹೌದು ಸರ್.
ಕಾಣಿಸ್ತಾನು ಇತ್ತಲ್ವಾ?
Yes.
Okay. For the time being we will stop and I will continue in